The following is part 1 of an excerpt from a Reddit post made by an individual claiming to have the full truth on what we call alien greys. In this online interaction, they even answered questions from people in the comments section. A link to the original post can be found in the description of this video. Make of this information what you will. From the late 2000s to the mid-2010s, I worked as a molecular biologist for a national security contractor in a program to study exobiospheric organisms, or EBO. The aim of the program was to elucidate the genome and proteome basis of these organisms. Although the study of OBCs has been going on for decades in other programs, the new high-throughput DNA sequencing technologies of the late 90s unblocked stagnant research in this area. Since then, several breakthroughs have led to significant advances in our understanding of the genome and proteome of these beings. What we've learned so far has enabled us to outline some disconcerting perspectives about our place in this universe. Briefly, we've discovered that the EBO genome is a chimera of genomes from our biosphere and from an unknown one. They are artificial, ephemeral, and disposable organisms created for a purpose that still partially eludes us. I'll be substantiating my statements after a brief introduction. The reason for disclosing these secrets is quite simple. I believe that every human being has the right to know the truth, and that to progress, humanity needs to divest itself of certain institutions and organizations that will probably not survive these revelations in the long term. I'm aware that I'll have very little impact in this regard, but I still believe that small leaks are necessary to break the dam of misinformation on this subject. When the governments will eventually reveal these secrets, there will undoubtedly be a societal upheaval. But in my opinion, the longer we wait, the worse it will be. I choose to divulge what I know anonymously out of selfishness for the well-being of myself and my family. I'm aware that this diminishes the reach and credibility of my message, but it's the furthest I am willing to go. I chose this forum because it offers a good compromise between anonymity and popularity. In order to protect my anonymity, I will be purposely vague or even contradictory about any information that could identify me: date, education, role, etc. I'll even introduce red herrings in this respect. I want to make it clear that any information related to the subject of the research will not be treated in this way. Before going any further, please excuse me if you find it difficult to understand what I'm explaining. Some parts of my text are very technical. It's difficult to find the right balance between vulgarization and scientific explanation. I'll continue by talking about myself. What's the point of talking about me knowing that the information will necessarily be misleading? I simply want to introduce a perspective on the type of people who work there, normal scientists. I have a PhD in molecular biology. I didn't actively seek to be part of this program. Rather, it was a stroke of luck that introduced me to one of the senior scientists. I met this person at a conference where I was presenting a poster on my PhD research. When I think back, I don't believe he was impressed by what I was presenting, because it was quite frankly a project that wasn't going anywhere. I think it was rather the most important aspect of a professional life: the attitude and the ease with which you make connections. Shortly afterwards, I graduated and received a call from this person offering me a position. At the time, everything pointed to me working in a regular laboratory. I did a series of 3 increasingly suspicious interviews, each in a different location, where my scientific background and knowledge became less and less relevant. The first was with 2 of the senior scientists, the second and third with people I've never seen again, and who were obviously not interested in science. Sometime after the interview, I was asked to go to a 4th location, where what seemed like a corporate lawyer presented me with an NDA. He made sure not only to explain every detail, but also that I understood the consequence of not respecting it. The first employment weeks were by far the most memorable, although I spent most of that time in a depressing archive room. It consists almost exclusively of reading about the subject of study and to get us up to speed. There's no secret Wikipedia or even a reference book to guide us. There are only dry reports, memos, presentations, procedures, and SOPs. These documents are almost exclusively about the biology of EBOs, but there are also a few that deal with other subjects such as their food, religion, or culture. There were no documents on their technology. As mentioned above, the aim of the project is to gain a better understanding of the EBO genome and proteome. To achieve this, a team of around 20 scientists, 4 senior scientists, and a director was involved. The scientists, like myself, had as their main responsibility to carry out the technical work. As each scientist had, to my knowledge, a PhD, we were all somewhat overqualified for what is ultimately a technician's job. The senior scientists, who make full use of their diplomas, had the task of designing the assays and had a supervisory responsibility. They were also in charge of training new employees and sometimes even came in to do technical work. The director, of course, was the person in charge who dictated priorities to the senior scientists. He was rarely on site, and the few times he was, it was to attend meetings. Other than the scientific staff, there were security guards working for one subcontractor or another. There were no support staff such as janitors or maintenance workers. Scientists were responsible for this kind of work. In addition, logistical constraints ensure that every scientist is capable of carrying out any technical activity. The laboratory itself is located in Fort Detrick, Maryland, in a building used for legitimate biomedical research. The clandestine operations are carried out in a restricted part of the basement, out of sight from regular workers. Contrary to what one might imagine, the biosafety level is not maximal for this type of research. Indeed, the lab containing EBO samples or derived cell cultures is BSL-3, while the lab where assays are conducted are only BSL-2. The BSL-3 area of the facility includes a freezer room and a cell culture lab and is only accessible through an antechamber from the BSL-2 section. EBO carcasses are preserved in horizontal freezers at a temperature of -80 degrees Celsius nominal. To maximize the preservation of these carcasses, they are preserved in vacuum bags, and the air in the room is controlled to minimize humidity. There are only 4 bodies, and none of them are complete. It's obvious that these creatures have died as a result of major trauma. I've never witnessed a motorcycle accident fatality, but it probably looks similar to this. It is acknowledged that there are more EBO carcasses at other locations. The cell culture laboratory, as its name suggests, is where cell lines derived from EBOs are grown and related activities are performed. I'll talk in more detail about these specific cell lines later on. The BSL-2 part is mainly used for assays, immunohistochemistry, genetic engineering, immunocytochemistry, storage, etc. There's also a cell culture lab, but this is used for more traditional cell lines. Other than the labs, there are all the amenities you could find in an office. Note that the internet access is limited to senior staff and up. There is, however, an intranet for bioinformatics needs. On the subject of the biology of these beings, I'll start by discussing genetics, then their gross anatomy, and finally their biological systems. For the sake of clarity, the information that I provide here is an aggregation of what I have observed and what I have read. I will make many comparisons with human anatomy because it is the most logical reference: genetics. First, I'd like to discuss their genetics. Their genetics are like ours, based on DNA. This fact was very puzzling for me when I first learned about it. We imagine that beings from an alternate biosphere would have genetics based on a completely foreign biochemical system, and surprisingly, this is not the case. Several conclusions can be drawn from this surprising revelation. The one that immediately comes to mind is that our biosphere and theirs share a common ancestry. They're eukaryotes, which means their cells have nuclei containing genetic material, which suggests that their biosphere would have been separated from ours sometime after the appearance of this type of organism. The term exobiospheric organism is actually a misnomer, but as it's a historical term, it's still used. Their genetics are not only based on the same genetic system, but they're also even compatible with our own cellular machinery. This means that you can take a human gene and insert it into an EBO cell, and that gene will be translated into protein, and this of course works in reverse with a human gene inserted into an EBO cell. There are important differences in post-translational modifications that will make the final protein non-functional, but I'll discuss these later. Their genome consists of 16 circular chromosomes. You're probably familiar with the concept of intergenic region or junk DNA. These are basically DNA sequences that don't code for proteins. These are evolutionary residues, transposons, inactivated genes, and so on. To give you an idea, in humans, intergenic regions represent approximately 99% of our genome. I'm aware that these sequences aren't completely useless. They can be used as histone anchors, as buffers to protect coding DNA from radiation, or even as alternative open reading frames, but that's rather peripheral. What's particularly striking about the EBO genome is the uniformity of these intergenic regions. We see the same sequences repeated everywhere, and the distance in base pairs between the genes is virtually the same throughout their genome. The result is a minimalist, highly condensed genome. In fact, it's much smaller than ours. Moreover, the quantity of protein-coding genes is even significantly lower than ours, probably due to genetic refinement, but also to biological processes that are absent in EBO. The uniformity of these sequences is a major indication of the artificiality of these beings. There is no complex organism on Earth that has such elegance in its sequences. There is no evolutionary pressure that can lead to this kind of characteristic other than genetic engineering. This concludes the reading of this excerpt portion from the Reddit post. What did you think of this story? Do you think it's real? Leave your comments below and make sure to like and subscribe to UFO Zone for a notification when part 2 comes out.